Hi, everyone. Thanks for the invitation. Today, I'm going to talk about a recent preprint investigating the immune microenvironment in pediatric CNS tumors and neuroblastomas, accessible through this QR code. Immunotherapy in pediatric cancers, distinct from that of adults. One approved agent is anti-GD2 dinutuximab for use in neuroblastomas in combination with GM-CSF and isotretinoin, first reported by the COG, showing the significant improvement in event-free survival over standard therapy. What's really exciting is a recent publication by Dr. Meisner and others showing the treatment of the first 4 cases with K27M mutated gliomas with anti-GD2 CAR T-cell therapy, showing the improvement in disease control and quality of life, really showing the novel, how this novel therapy might help these patients. Of course, the number of clinical studies for pediatric brain tumors that involve some form of immunotherapy has increased in the last 5 years compared to the 5-year period prior. And these involve a number of immunotherapies ranging from vaccines and immune checkpoint inhibitors to viral therapies and CAR T-cell therapies. And given these ongoing clinical studies, there's a need to better understand and identify a robust biomarker to predict and select patients that may respond to these treatment modalities. There are four major classes of predictive biomarkers for immunotherapy. One is measuring the molecular targets. Examples include B7H3 or PD-L1 by IHC or by RT-PCR. Tumor intrinsic features like TMB that's already been improved in adult cancers. There are currently 2 trials that children over the age of 12 with TMB high cancers are eligible. Microenvironmental factors, composition, functional status of immune cells, their interactions with stromal cells and tumor cells. That's another class of biomarkers that actively being investigated. And of course, not to forget the host factors, gut microbiome, peripheral T-cell repertoire, and germline variants and HLA polymorphisms in children's cancer. In this study, we focus on looking into the patterns of immune infiltration in tumors. And to study the patterns of immune microenvironments in pediatric CNS tumors and neuroblastomas, we compiled a cohort from 3 consortia, CBTN and Target, which were harmonized by Kids First Data Resource Center at CHOP, and data from ICGC collected and curated by the Kids Group in Heidelberg. This collection collectively included 925 cases with treatment-naive CNS tumors and neuroblastomas, spanning about around 12 cancer types. We used data from 79 PDX models collected by the ITCC project. Very briefly, we have curated a set of immune-specific genes and used consensus clustering to identify major patterns of immune infiltration. One cluster consists of 90 samples that shows the highest enrichment of 6 immune gene sets across the pediatric cohort. This cluster consists of MYC-like ATRT, craniopharyngiomas, and low-grade gliomas with BRAF V600E mutation. The second cluster seems to have a depleted lymphoid cell gene sets while showing some enrichment in myeloid group. We call this myeloid predominant to account for the possibility that tissue resident microglia might overlap with the signature in monocytes. And this cluster consists of some neuroblastomas, but also meningiomas, craniopharyngiomas, and low-grade gliomas with BRAF fusions. Of note, the subset of this group with expression of genes involved in mast cells and macrophages also show the expression of FCGR1A that's shown in the report by Dr. Avi Mayan earlier today. The third group, we called it immune neutral. About 42% of these cases do not show any significant enrichment or depletion of these gene sets. And lastly, this cluster shows the depletion of all these immune gene sets. This consists of about 18% of the entire PTNST, and includes sonic hedgehog-like ATRTs, medulloblastomas, and epidiomas. So really four clusters spanning 2 dimensions of lymphoid and myeloid cells. We asked whether cases in more inflamed clusters may have higher TMB, similar to associations in pediatric cancers, in adult cancers, and TMB as determined by non-synonymous SNVs or SNV plus indel was not independently associated with any of these immune clusters. In high-grade gliomas, however, there is a significant association where with cases in myeloid predominant shows higher level of TMB compared to immune neutral or excluded. That's probably driven by a few cases of hypermutants and biallelic mismatch repair deficiency. We recovered TMB cell repertoire clones using TCR and BCR reads found in the RNA-seq. Using the hybrid capture of TCR gene segments from the same RNA-seq libraries, we confirmed the TCR diversity estimates from RNA-seq could be representative of the actual diversity that's shown here in the x-axis. TCR diversity was significantly higher in pediatric inflamed and myeloid predominant compared to immune neutral or immune excluded in both neuroblastomas and CNS tumors. So there's a positive correlation between the estimated diversity from the RNA-seq and the total number of TCR reads, because the TCR coverage is very shallow in RNA-seq. However, there is a subset of these cases that shows an outlier diversity, suggesting a highly polyclonal repertoire or a clonal expansion in samples where diversity is lower relative to their number of TCR beta reads. So 2 representative repertoires here shown as the circle plots. And so we can use this sort of linear regression to regress out the effect of TCR reads on the Shannon diversity, and would get at some measure of TCR diversity that's controlled for the number of reads recovered. So if we do that and look at the residuals, we see that there's no significant T-cell gene signature enrichment as expected. That's controlled for the TCR reads. But we see that the samples with lower residuals corresponding to a clonal repertoire would have a higher level of dendritic cells, higher level of B-cell clonality as determined by gene index, and higher expression of immunoglobulins, suggesting the clonal outgrowth of T cells in the presence of interactions with antigen-presenting cells. Lastly, to validate the lymphocyte infiltrate levels across the 4 microenvironmental clusters showed you Dr. Aldrich at CHOP performed a tissue image analysis using the published deep learning approach for CNS tumors from CBTN, where we had the matched H&E slides and RNA-seq data. On the right, you see the representative images of 3 cases with 1, 10, and 15% TIL score. The samples in pediatric inflamed showed a significantly higher TIL score compared to myeloid predominant or immune excluded. So in summary, what I'm showing you is general patterns of immune microenvironments across pediatric cancers, covariant immune cells in treatment-naive CNS tumors and neuroblastomas. These patterns appear to be independent of TMB, at least in the non-hypermutant cancers. What we hope to further address as more data is becoming available is the evolution of immune microenvironment under treatment pressure or as a natural progression of the disease. We see some immune infiltration, as I've shown you, and some level of clonal outgrowth in a subset of these cases, but we have not yet found a source for possible neoantigens. That's something that we'd like to address with a bigger data set. And of course, the effects of both germline variants and HLA polymorphisms on immune microenvironment is largely not known. Something that typically needs a bigger data set to be able to address. And so to address some of these questions, we now have an active public workspace on Kabatica for immunogenomic data sharing and analysis that's funded by CBTN. Our process data and output files are deposited here and are accessible to everyone to reuse and reuse. We continue working on pipelines and processing more data as they're becoming available, and hopefully form a resource for the immunogenomic community to work on. And lastly, again, another sort of collaboration with CBTN, how we are using resources that's provided by CBTN, is funded by Alex Lemonade Stand Foundation. We try to address how immune microenvironment changes over time in low-grade gliomas by single-cell RNA sequencing of serial samples requested and obtained from CBTN. And this is part of the single-cell pediatric cancer atlas that we share the sequencing data in real time with CCDL and forms a harmonized data set that increases the power for our study. I'm on time, great. I would like to thank Trevor Pugh, my mentor, and Stefan Pfister for supporting and collaborating on this study. My postdoc and this study largely are supported by the Terry Fox Profile Program in Canada and Princess Margaret Foundation. I would also like to thank CBTN, Kids First Data Resource Center, and Adam Resnick for supporting this group, this work along the way, both financially and technically. My trip here was kindly funded by CBTN, so thank you. And of course, special thanks to patients and families